Have you ever purchased a box of strawberries, placed them in your refrigerator, and come back a couple of days later expecting to eat delicious, firm strawberries, but much to your disappointment, you find that most of them are actually overripe? You might as well just eat jam. Just how does ripening happen anyways? When a fruit ripens, pectin polymers are solubilized and depolymerized, as are the matrix glycans, and neutral sugars are being lost from the pectin side chains. These events all happen at once. The precise mechanism for pectin depolymerization is still unknown, and researchers are using tools such as Antisense technology to find out which genes play a role in fruit ripening, among other agricultural functions. To understand Antisense, we must first go back to the central dogma. Replicated DNA is transcribed to pre-mRNA in the nucleus, which is then spliced and processed into mRNA, or messenger RNA, and exported to the ribosomes, where it is then translated into a protein. Protein synthesis can be stopped at the transcriptional level, for example, by introducing a frameshift mutation, or at the translational level, using, for example, antisense technology. Antisense technology takes advantage of base pairings and uses complementary strands to bind target mRNA. Antisense can be employed using different mechanisms, such as ribozymes, RNA interference, aka small interrupting RNA, and microRNA, or antisense oligonucleotides. Ribozymes are enzymes that cleave the phosphodiester backbone of RNA by nucleophilic attack on the 2' oxygen, forming a 2'/3' cyclic phosphate and a 5' hydroxyl product. This inactivates RNA, destroys its message, and prevents protein from being made. There are several types of ribozymes. Some examples include RNase P, which acts on transfer RNA and uses zinc and magnesium as cofactors, hairpin ribozyme, which does not require metal cations and helps process RNA by cleaving and end joining, and hammerhead ribozyme, which also cleaves and ligates RNA. RNA interference also leads to RNA degradation, but does so in a different manner. RNA can be divided into two types, the coding and non-coding type. Coding RNA is messenger RNA, and the non-coding RNA includes transcriptional RNA, which is ribosomal RNA and transfer RNA, and also small RNA, which consists of small interrupting RNA, microRNA, small nucleolar RNA, and small nuclear RNA. Dicer, or in the case of plants, dicer-like enzyme, first cleaves the double-stranded RNA into uniform 21 to 25 nucleotide long fragments to create siRNA or mRNA with 3' overhangs. RNA-dependent RNA polymerase is recruited, triggering and amplifying the silencing effect by generating more double-stranded RNA. siRNA or mRNA then binds to an aragonite protein, which is part of the RNA-induced silencing complex. The RISC helicase enzyme cleaves off the sense strand from the double-stranded RNA, and the remaining strand, the antisense strand, is now free to bind to target mRNA. Once bound, and if it is a perfect match, the RNase H enzyme, slicer, cleaves the mRNA, and once again the message is destroyed and protein cannot be synthesized. If the antisense strand from the siRNA or miRNA, however, binds to the mRNA, but there are mismatches in this duplex, the mRNA is not cleaved, but translation is physically blocked by RISC. Basically, RISC is stuck, and this causes translational arrest. MicroRNAs are 21 to 25 nucleotides long, single-stranded, and are transcribed by RNA polymerase II. They are partially complementary to mRNA. Each mRNA molecule has miRNA binding sites. We first start in the nucleus, where double-stranded hairpin loops, called primary miRNA, are formed. They contain a 5' cap and a 3' poly A tail. Then the RNase 3 enzyme, DRASHA, and the double-stranded RNA binding protein, GGCR8, further process them into approximately 70 nucleotide-long preliminary RNA, which then gets exported to the cytoplasm via exportin 5. Once in the cytoplasm, dicer further cleaves the pre-miRNA into 21 to 25 nucleotides long. miRNA then goes through the same RISC process as described previously. Antisense oligonucleotides are not naturally occurring and are synthesized to target the mRNA one wishes to study. In order to make the oligonucleotides, one must have the sequence of the gene one wishes to study, meaning this technique cannot be used for a novel or unknown species. There are 3 approaches to antisense oligos. In the first generation, complementary 15 to 20 nucleotide long oligonucleotides are made and bound to the target mRNA. This binding prevents translation through either physical blocking or through recruitment of RNase H, which recognizes the RNA-DNA duplex and hydrolyzes the RNA. The oligos are then released and combined to other RNA. However, although they do have high affinity for RNA, they are susceptible to nucleases, which does not make them a very stable long-term option. In the second approach, the oligos are less susceptible to nucleases because the phosphodiester backbone is modified. First with the phosphorothioate and then using 2' MOE or methoxyethyl. They prevent protein synthesis only by translational arrest. RNase H is not recruited in this approach. Although the oligos are more stable using this approach, they have less affinity for RNA, meaning higher molar concentrations must be used, and they still have short half-lives. The third approach combines the previous 2 approaches, creating gapmers. Gapmers are antisense sequences that have modified 2' ends but also have regular centers. The ends protect the oligos from hydrolytic cleavage, and the centers recruit RNase H to degrade the mRNA. Once degraded, the stable oligos are free to bind to other mRNA. This approach has increased RNA affinity compared to the second approach, and is more effective than the first approach due to its immunity to nucleases. Translational arrest and mRNA degradation using antisense strands is a valuable tool that can help determine the function of genes, not just for therapeutic purposes, but also for added agricultural values. For example, going back to strawberries, in 2015, Dr. Paniagua in his lab used antisense technology to show that novel FAB-Gal4 beta-galactosidase gene was a key player in fruit ripening. It has been hypothesized that pectin disassembly is caused by B-Gal genes removing carbohydrates from RG1 polyuronides. Dr. Paniagua isolated FAB-Gal4 gene from a full-length cDNA library. He isolated the RNA and amplified using quantitative real-time PCR. He then cloned antisense FAB-Gal4 by inserting a 300 base pair FAB-Gal4 gene into a pCR8 GW topo donor vector in the antisense direction, and then recombined this vector with PK7WG2 acceptor vector. The vectors were then transferred to the actual strawberries via electroporation. The silencing of the FB-Gal4 caused strawberries to be significantly firmer compared to the controls, and they also increased the sugar levels while decreasing the pectin solubilization. So next time you eat an overly soft strawberry, you can thank the functional FAB-Gal4 gene.